Hello, everybody. Thank you for sticking around. For those who are still here, I recognize a lot of these faces have been here since the morning, so thank you for sticking around the entire day. We are here now for our final panel, Cell to Satellite. This is one of the areas of New York space ecosystem we were not super familiar with when we got started, but have become much more familiar with as time has gone on, because there's a lot of really interesting things happening here under the umbrella of quote unquote space medicine. And with that, I will turn it over to the actual experts. Starting off with Shishir.
Thank you, everyone, for hearing. Great. Thank you, everyone, for joining us today. We're going to be talking about everything that's related to biology in space, medicine in space, why that even matters. Like a lot of you here, you may not have been familiar with, what the next wave of innovation in space looks like. And so here I am with, three of those, minds that are really shaping what, that next new economy in space and space utilization looks like. So, I'll do a quick intro and then let's jump into what the meat of this conversation is. With us is Dr. Chris Mason, who's a professor at Weill Cornell, director of World Quant. His pioneering research spans genomics, space biology, diagnostics, with over 350 peer-reviewed publications, patents, multiple biotech ventures. He's also the author of The Next 500 Years and the Age of Prediction. If there is a study in space, one way or another, Chris is involved. Next, we have Jeremy Wain Herschberg. He's a research specialist at Weill Cornell, director of operations at ChemBank, with a strong background in diagnostics and space biospecimen logistics. He has supported over 15,000 biospecimen collections across 8 space missions. He's also involved and conducts multi-omic research and plays a key role in advancing space medicine infrastructure. And then finally, we have Dr. Devon Lundine, who I know very well. He's a postdoc research fellow at Memorial Sloan Kettering Cancer Center here in New York. He specializes in DNA repair. He's a winner of the 2023 Axiom and Boryung Humans in Space Challenge. And that's really where I met him. We also won. And he contributes to multiple NASA programs and holds a PhD from City University of New York, where he studied DNA replication in breast cancer. And then finally, I am Shishir Bankapur. I'm the CEO of Helogen. We are a space biology and infrastructure company headquartered here in New York. Our real goal is to build the infrastructure and the ability for teams and researchers to bring breakthroughs in medicine and materials to market faster than anyone else. Our hardware really enables early stage drug discovery using the conditions of space and manufacture of next generation of biomaterials in space. Our first launch on was in November 2023 on Transporter 9 with SpaceX. And we have a bunch of upcoming satellite missions over the coming years. So to begin, I think, let's start with why space. Each of you works in biology, works at the intersection of biology and space. Why is space not just a backdrop, but really the frontier of for the work you're doing? Let's start with you, Chris.
Sure. Okay, great. Well, first, thanks for having me and thanks for the introduction. And it's really nice to be here today and to see a lot of people, met a lot of really fascinating people already today. And I think for why space, I think there's many reasons. I mean, one of them is just its inspiration to see children to look up at the sky at night and imagine that the future is brighter than the present, which they get some degree of hope. Space is also technically challenging, which is great from a manufacturing, technology, and development perspective. It forces you to be efficient with power, essentially with size, with infrastructure, with usability. So when we plan missions and do a lot of work for collections for the crews, it has to work. It has to be easy. It has to be very, low power, low mass, ideally. So it's a great forcing function. And then also why space? Because as mentioned, you mentioned in my book, the next 500 years, at some point, the sun will engulf the Earth. And I think about this every morning. And I think, well, we've got to go that way eventually. So at some point, we've got no choice. Do you want to?
So you mentioned a lot of good things. Excuse me. I'm fighting a cold. I'm over it mostly.
You should take a sample and take a look at
it. You mentioned a lot of good things there. So I want to open it up to, the other two. What can we do in space that we can't do here on Earth, just in terms of the work you're doing?
Well, I think that fundamentally space research is unique because of the extreme nature of that environment. And some of that, I would say that environment is characterized by two major things. One is microgravity and the other being radiation. One of those is a decent bit easier for us to recreate on Earth, being radiation. We can certainly do some microgravity studies, but creating a prolonged microgravity environment is not that easy to do on Earth. And so that's a particularly unique opportunity. When it comes to recreating radiation environments here on Earth, frankly, when you're studying a system, you'll often see, in a lot of cases, a large response when whatever variables you're tweaking are dialed up. Thankfully, we have guidelines that prevent us from just studying radiation by putting a human in a high radiation environment. That's not something that we think should be allowed. And so in this age where we are sending plenty of people to this environment, I say, even not being a part of the decision to send these people, there are larger powers that be that have decided to send these people. And there's people who have said, I want to go. It's our opportunity, if not our duty, to try to understand what's happening to these people's bodies when they go to space.
Devon?
Yeah, I just wanted to tap on that as well. I think that's a great point. Both made great points. One part of it I think that is very impactful too is not only this uniqueness of the spaceflight environment, but also this idea of trying to bring back things that to Earth that we're either not able to make on Earth. Many companies are doing in-space manufacturing, things that are benefiting from microgravity. But also this idea, at Memorial Sloan Kettering, we work in all types of specialized cancers. And the idea of having kind of a chronic low-dose radiation in space, there's a lot of crossover between what we're doing on Earth to treat patients and what's going on in space too. So there's a lot of kind of back and forth that, one thing that you do on Earth helps in space, and things that you do in space can kind of come back to Earth terrestrial bound helpful products and things like that.
You mentioned something, or you touched upon, a line of thinking that, I've been thinking about a lot at Helogen, which is what can we do in space that, ultimately benefits the economy here on Earth, whether, we're taking advantage of accelerated aging. For instance, there's plenty of research that points that certain kinds of tissues age like 30 times faster in space. There's organoids that, form faster and are more stable in space. You mentioned, there's no access to chronic low-dose cosmic radiation here on Earth. What does that mean for applications, and what does that mean for human health on Earth? Let's
Yeah, and I think the radiation example is a great one for radiotherapy. So, for example, if any of you have ever had brain cancer, hopefully none of you have had brain cancer, but if you have, there's a kind of therapy called gamma knife, where you make these planes through the brain where you want to actually get, the tumor's only maybe this big. And there's no way, if you drilled in with a linear dose of radiation, it would kill all the tissue on its way. But with a gamma knife, you can actually do slices of radiation and figure out a way to make it so that the center of all these slices is where the tumor is. And the other parts of the brain, though, for example, get this low dose radiation. And we have very few ways to, prevent damage to other tissues, at least historically we have. And so we have to say, well, that's just that, that comes with the territory. If you want to get rid of the tumor, other tissues will be damaged. But the big danger is then they get additional mutations. They have their own long-term risk of carcinogenesis. It could disrupt the neighboring tissue, could lead to metastasis, or all these questions in cancer and radiation and radiotherapy that have been around for really 30, 40, 50 years in some cases. We can now, because there's been this focus on radiation and prevention, some of the newer tools that we've recently flown actually on the FRAM2 mission, which was just a few weeks ago, we're working a lot in the lab of taking some of these old questions in radiotherapy and saying, okay, how can we address them, and then use genetic tools to actually reactivate genes that can help with radiation resistance. And so Jen-Kun Kim's a postdoc who's in our lab who has been tweaking cells. So imagine when you walk into a room and you turn on a light switch and you don't think twice about turning something on or off, or you go into your kitchen, you can turn off an appliance. We can now do the same thing with very specific genes inside of cells using variations of CRISPR systems called DCAS9. So you deactivate the cutting part of the enzyme and just have it still have the homing beacon function. So this is something that when I was a graduate student and undergraduate, I thought, oh, what if someday we could control cell specificity and turn on genes at whim, which was complete fantasy until really the past 5-6 years, where this is now being actively pursued in our laboratory and many other labs. So that's one component. But then another thing is, modifying existing gene profiles with human cells is one avenue we're pursuing, and many other labs as well, but also taking genes from tardigrades is another thing that we've been doing, and modifying human cells and yeast cells to have radioprotective genes that came from a different species and plop them into human cells or other yeast cells, which has been really extraordinary to see the efficacy of, effectively almost an alien protein inside of a human cell that confers this radioprotective ability. And that was something we've been working on, but then a paper just a few weeks ago was published actually taking away, at least in mice, of actually using sort of nanoparticles to transmit these D-sub proteins, these tardigrade proteins, and give this radio protection to other parts of the body while you're targeting radiotherapy at one spot and protect the rest of the body. So this era of learning from other creatures, ways how they survive radiation and high dose radiation, and then trying that in human cells is now upon us, which I think is very exciting. And that's one factor. Then also just the long-term effects of radiation. We think about lifelong risk of cancer, something else called clonal hematopoiesis, where we all carry mutations and they change over time. And we can see that space here is a forcing function. Telomeres change. They get a little bit longer when you're in space, but then they get right back down when you get back to Earth. And actually, if you look closely at telomeres, they are more damaged. So we know there actually is a lot of stress on the body. And some things look older, accelerated aging, like you mentioned. Some things look a bit younger, like longer telomeres in space. But we know it's a lot of stress on the body, and so we're just learning about all of them, and then applying them to Earth. Is now we can take some of those same tools and metrics, and watch cancer patients, watch people who may be 5 or 10 years away from leukemia, but see if they're on that trajectory, and start to track those things. There are examples from.
Yeah, I think one element of doing space medicine is just how remote the environment is of space. And so a lot of the work I do with the Cornell Aerospace Medicine Biobank is the logistics of collecting, processing, and stabilizing samples that ultimately we'll study to try to learn what's happening to the body when someone's in an environment as harsh as this. And so, what that means is coordinating with these commercial space companies to rendezvous with their crew, wherever their crew happens to be, and organizing a lab, having a collaborative lab nearby wherever they are, because some of our most sensitive samples, the collection method requires that tube be processed within a couple hours of collection in order for it to fall within the use case of that medical device. And so that means we're traveling to a location, we're coordinating with a lab so that we can be there to process and stabilize these samples. And frankly, there are places on Earth that are very remote and that medicine is still required. So I recently went to a meeting on remote emergency healthcare in places on Earth and how space can be applied to that. There are, let alone mountain climbing or something like that, where you're completely out in the wilderness and might need treatment. There are rural communities that are, serviced by a single ambulance and don't have a hospital. And so the way that we're studying these astronauts ultimately is going to inform the way that we bring healthcare to people on Earth also.
Want to add something to that, Devon? That's great. Chris, you mentioned, we're able to, edit genes or manipulate, all of those things based on research that's been built so far. For those of you who don't know, Chris was involved with the NASA twin study, which was a landmark investigation in understanding what the effects of long-term spaceflight are on the human body. So we had, a twin, Scott Kelly and Mark Kelly. Mark remained on the Earth, and Scott Kelly, as you all probably know, spent about a year in space, and we tried to understand, what the differences were, based on his experience. Chris was a principal investigator on that study, and contributed significantly by analyzing studying the genomics, epigenetics, gene expression, and the sort. So based on all of that research, what were some of the findings that you think is most critical to upcoming missions and deeper space missions as we're shifting priorities from maybe Moon to Mars?
Yeah, and Moon to Mars is, very likely still going to be the catchphrase for NASA's plans to get eventually to Mars is the long-term goal. Elon has been tweeting out, or he's been Xing. What's the verb if you're Xing? Does anyone know if you're still tweeting? Does anyone, anyway, when Elon was Xing, which sounds kind of gross. I don't know what Xing is, but it's I don't know what happens. But he has said, we can have uncrewed mission, maybe with Optimus robots by within 2 years, maybe crewed landings within 2 to 3 years, which is wildly ambitious. But even if it's 6 or 8 years away, that's still really close. So it's an exciting time. But the lessons from the twin study, so was there had been longer missions by the Russians. So we know that the human body could live as long as maybe 400 days in space was known was possible. But there had never been a study, a really deep dive of genetic, molecular, and cellular changes, what happens. And that's where we first saw the longer telomere result in space, which again, on average is longer, but we also see a lot of damaged telomeres. So there is DNA damage we can observe. We can see calcium coming out in the urine. We can see damaged DNA coming out, sort of eluted out of the body. And we could, see a lot of stress on the immune system. Those are some of the big factors we saw. But we did do a vaccination in space in the middle of that year-long mission, and the T-cell diversity was actually maintained, and B cells were able to function. So we think it's not that the immune system is non-functional. It's just that it's very stressed. It seems like it's wildly activated generally, and significantly more so than on Earth. And that was with Mark and Scott Kelly, and of course now it's Senator Kelly, and also now Jared Isaacman was the PI for one of the PIs for the Inspiration 4 study. So now we have maybe the NASA administrator and some senators, DNA, and the biobank. So we're slowly collecting samples from all of the government, and we'll tell you next year the difference between Democrats and Republicans. We'll tell you, and it's just a couple of snips probably. We don't know, but we ongoing collections done. Actually, to Jeremy's credit, it is the largest biobank, one of the largest in the world, but of its kind, it's by far the largest. It has over 15,000 samples from several missions, including follow-on studies for the twins. And that was, NASA's first jump at it, but of course it was also, laughably underpowered. We only have 2 people and only really 1 in space. So there's only so much you can learn. But when you do a lot of longitudinal sampling, we did 23 collections over the course of 3 years before, during, and after spaceflight. What you lack in statistical power, you can get through a lot of longitudinal sampling because you build these really clear baseline profiles for every biomolecule. And that's been very helpful to get as much, blood out of the stone of the mission and learn as much as we can. But I think, it was NASA's first foray. But what's interesting is that was the bedrock for the SpaceX Inspiration 4 mission, the Polaris Dawn mission. We've released all those protocols, all the tools. And so what's really exciting is other groups are now taking a lot of these protocols and these methods and putting them to their studies. And there's a wide range of space stations being planned. There might be as many 4 space stations in orbit in maybe 3 or 4 years, which is pretty exciting, and large. A lot of them are really large. And there's Chinese plans, Indian plans, and obviously US and European plans to go to the Moon and Mars. really soon. So it's you're excited to have to learn more about the body in deep space. And the take home, the TLDR is it looks like the body can survive, and it's really stressed out, not necessarily happy, but actually coming back to Earth was much harder on the body than leaving Earth in terms of the stress on the inflammatory markers and just the stress on the body. And, it just the inflammation, the rashes, really was rough on the body for almost every crew that's gone up. The longer you go up, the harder it is to land.
Thank you so much. Just a follow-up on that. So based on everything that, that we learned, by sending people in space or keeping them and observing the effects of space on their bodies, what can we take away to, fight or better health here on Earth?
Good question. So that was always one of the main goals of NASA and other space agencies is take lessons from space and bring them back to Earth. A lot of it was we did some of the first nanopore sequencing in space, actually right after that mission and some of the protocols during that mission. So that lets you do sequencing. When the pandemic hit, we could do portable genetic sequencing because we'd already miniaturized those protocols a lot during that mission and some of the follow-up missions. So I guess more remote diagnostics, as Jeremy was discussing, is that the ability to go anywhere and do medicine wherever you might be, and point-of-care diagnostics was helped by that. Actually, interestingly, some of the same stress markers we observed showed up again during COVID. We could see some of the same, like IL-6 and other markers that showed up for really severe immune stress popped up again. And actually, some of our first emails at the hospital looking at COVID patients was weird. It looks just like we're looking at astronauts, but they're people in the hospital. So interestingly, some of the immune stress in spaceflight gave us already the same targets that we could see to use for COVID and probably other infections as well. So sometimes stress on the body in one context helps elucidate and give you new targets of what could be useful for another virus, which is what we've seen.
Amazing. So I'm just going to move over to Devon real quick. You won the 2023 Humans in Space Challenge based on, the research that you were doing at MSK. Can you talk a little bit more about that and, what the implications are for both space health and its applications here on Earth?
Yeah, for sure. I think to piggyback off of things that Chris said, I think the twin study for us was really this landmark study for us, for many labs, for all the researchers across the world. And we were able to take a look at that data and essentially see the effects on DNA and the genomic scars that were kind of left over from the twins as a result of the year in space flight. And we're DNA repair experts. We're not necessarily space experts, but we've been now trying to kind of jump into the fray. And so the Axiom Boryung Humans in Space Challenge was our first kind of jump into the space world. And some of the lessons from the twin study include that particular types of DNA repair are modulated. So some types of repair are turned on, some types are turned off, some are more heavily used. And so this is true in particular cancer patients as well, and certain treatment modalities, certain kinds of radiation. So there's a lot of similarity between cancer patients on the ground and our astronauts that fly. And so we've been taking a look at drugs and other potential countermeasures as a way to kind of transiently modify the human response to radiation. And so, some approaches to maybe do gene editing or gene modification or introduce mRNA. We're still kind of a lot of interesting bioethics to kind of tackle around that. We're still kind of the mind of going back to classical kind of pharmacokinetics and trying to look at small molecules and drugs to essentially tweak the DNA damage response. And so that was originally our idea for Humans in Space. And I know that we'll talk about this a little bit more, but we leveraged all the great actually radiation infrastructure that New York State has to offer, which is some of the best in the world, to really test this. And so we've been able to—
Like Brookhaven or things like that?
Brookhaven National Lab, as well as the Columbia Neutron Beam Facility. They have a RARAF that's upstate, I guess. They're in Irvington. It's not, it's 20 minutes north of here, not really upstate, but so facilities like that, which are really best in class to try to address these questions and expand our statistical analysis, right? So we can do this on cells, we can do it on animals, on the ground, where we can basically have, an N of as much as we want, a sample size as big as we want, tested on millions of cells, in that case. Now, the translatability of that is tough, right? We can't necessarily, like Jeremy said, we can't put somebody in front of a particle accelerator just yet. But there is also the New York Proton Center here in New York, where we are putting people in front of a particle accelerator for cancer radiation therapy. And so there's all these things in, at least in the radiation world, which is what I'm most interested in, that kind of coalesce around this idea of, you know, we've got humans in space, we've got people on Earth. How do we kind of bridge this gap and make use of the tools that we have and continue to develop things like countermeasures that are either existing drugs or develop new drugs that can kind of tackle some of these major problems.
What was the thing that you won with Boryung? The prize was to do like more of the characterization or more screens, or what was the prize?
Yeah, so we got some pilot seed funding from Boryung and Axiom. So we were able to actually do our ground studies at RARAF and bring cells over and shoot them with space lasers, essentially a particle beam that simulates radiation. It's really hard to do the low dose that you see in low Earth orbit. So what we're able to do is kind of a Mars dose of radiation, and it's kind of as good as we can get for the high energy spectra of space radiation, and try to say, okay, do we see a reduction in DNA damage if we test these different countermeasures? And so far we have. We're still kind of in a preliminary phase, but we're scaling up to things like taking donated whole blood and trying to irradiate that and trying to get closer to this translatability question of, we've seen that this works in cells. Does it actually work in human adjacent or animals, or hopefully eventually we can test it in humans. So we'll see. The more people that you get in twin and octuplet studies, I'm sure that we'll be working together to try to solve some of this problem with the radiation.
Take triplets, quadruplets. If any one of you have in the audience clones, basically, let us know.
So Devon, as I understood and what I heard, is that the outcome of your work and your research is really looking at novel therapeutics, novel drugs, and novel countermeasures that wouldn't have existed otherwise.
Yeah, in some cases that's true, and in other cases we're taking this approach of taking existing drugs and applying them in a new way or ways that people haven't thought of before. And like Jeremy said, the uniqueness of the space environment is such that it takes very creative minds, I think, to try to repurpose or navigate around some of these tools that we have, or understanding what tools we need to develop. And so we're in that space of trying to do both.
Got it. Jeremy, quick question for you. You've been involved with collecting and analyzing all these samples and creating this sample bank. One of the things that is super interesting, to us and the space medicine world in itself is, are we seeing any breakthroughs or patterns that would be applicable for either space health immediately or Earth-based diagnostics?
Yeah, it's a great question, and whenever and it's one that comes up often in this line of work, and I always kind of lean back towards the direction Chris was going regarding statistical power and how important it is to qualify the things we're seeing within the context of our sample size, say. But I'm actually really excited about that because it emphasizes that we're really in the early days of building a massive database that's going to span across different layers of biology. So we do multi-omic research, being that, like, to take it back to cell biology without getting too complicated, every cell has DNA in it. And if you're studying that DNA, you're studying the genome. That DNA is transcribed into RNA. So if you're studying RNA, you're studying the transcriptome. That RNA goes on to make proteins. If you're studying the proteins, you're making proteome. And each one of these areas of study has classically, been thought of as its own area of study because it required its own set of tools in order to take that on. And so we're entering a new age of precision medicine at a similar time that we're entering the second space age, where we see, massive increase in the amount of crewed missions that are going to space. And we're trying to take each of those layers and consider them together as a whole picture in a way that we're relatively in early days in that regard. And so in regards to the biobank, we start piecing together this picture as we continue to collect more and more samples, and then pair those samples with publicly facing data. So if we have data that's published, someone says, oh, this is really interesting, I wish they ran this test on it, they can actually reach out to us, and we have copies of that same sample that then they can take, run whatever test they find interesting to do, and then publish that along with the data that's already there. And then that triggers someone else to have an idea of studying something interesting, and that catalyzes a lot of research. And so I think that, we're building the database, and all of the findings that we're seeing are remarkably interesting as a snapshot of what happened to this individual when they went into a really harsh environment. But ultimately, the massive value that's going to spin out of this is going to take some time. But I think as it continues to come together, we're going to see what will be illuminated is larger aspects of systems functioning. So not just what's happening to your DNA, but how is your body responding to these harsh environments? And that's when I think we'll really see the larger medical outcomes that are going to be applicable to people going through something that might be similar on Earth, like you brought up aging. In space, we see people's bones break down and their cardiovascular health declines because your heart doesn't need to pump against gravity, your weights, your bones don't need to hold you up, and it's entirely possible that as we understand the underlying cellular mechanisms that are driving those changes, those might be the exact same driving factors as to why you see osteoporosis in aging populations. And so, I think that we're not quite at the point where we're able to make those larger conclusions, but we're building that database, and it's on the horizon, and it's going to come quicker than hopefully any of us are actually anticipating.
Yeah, and like sarcopenia or other sort of wasting disease, cachexia is another sort of disease where you see wasting, and some of those markers show up also in spaceflight. So then we can see other just signs of aging, really, and frailty. We can pick up in spaceflight and learn about them faster potentially. Yeah.
Oh, that's great. Jeremy, you actually, touched on the ability to, start, or rather we're on the horizon of commercialization. So, one of my big interests through Helogen is, we want to get people there faster. Typically it takes, a couple of years to get to the ISS. Helogen, we build our own small sats with multiple labs on there. So that we can launch, faster, high cadence at a much lower, price point as compared to, some other things. So as we're starting to look at commercialization and commercializing some of this research, I have a question for you, Chris. You're the founder of, or you're the chairman of BioAstra, with the goal of, taking everything that we've learned with the, 15 years of the ISS National Lab being, a thing, and applying that to, commercial applications, be it, drug discovery, be it, understanding, diagnostics or space health. How do you view, where we are at a point in time? Are we at the right point to try and take on some of those efforts, or are we a couple of steps behind?
I think if you'd asked me 2 years ago, I would have said, it's a little early because the Starship hadn't really successfully flown, hadn't been recaptured. Some of these heavy lift rockets, New Glenn, were still in early stages. So I think now, though, so BioAstra is the goal is to really try and bring as many of these planetary tools and methods that are here on this planet, but get them out into medical tools, algorithms, really implementation that could help us survive on the moon, Mars, and anywhere beyond it. So that includes actually planning missions that would do an additional twin study. So we fill a Starship full of about 20 twins, and then you can also take clones of your own cells and make induced pluripotent stem cells. So we'd fly not just dozens of twins, but literally thousands of twins, at least in terms of the mini clones of yourself, which you can do as organoid models. You've maybe heard about these, but they're little derivations of your own cells that can serve as little avatars of yourself. So we want to fly a whole Starship full of those in the near future, and even have it do a mission that would go to Mars and Venus and back again, what's called the Twin Astra mission, which we published last year. So what's interesting is they were all hypothetical 2-3 years ago, but now the rockets are enabled. A lot of the biomedical tools have been at least piloted, But what's still missing is a way to integrate them all together and also have life support systems that could enable you to live in space for 18 months. If you're on the space station, there's actually a lot of infrastructure already there that we have to basically miniaturize and put into other stations and other spacecraft. So BioAstra is a nonprofit, though. We debated about, is this a for-profit, a nonprofit? I've founded several companies over the years. If you have a very clear for-profit goal, it's great to go after, but then you immediately are competitive to anyone who's close to that. By definition, they are your competitors in most cases. But so we wanted to make a nonprofit as kind of a clearinghouse and an open engagement with the entire community, for-profit, nonprofit, academics, governments, work with anyone and everyone to get all the tools annotated, cataloged, and start to get them integrated. So when you're going into new space stations, new missions, there's a place to visit them all. And also we have summer fellows that come every year that actually learn, that have very specific focused projects like pharmacogenomics and spaceflight, or hardware for imaging. So a lot of tools and methods that we have every summer. We have a summer fellows program as well. But it really just started because we couldn't find the catalog of every piece of hardware that had ever flown in space. And we thought, why the hell doesn't this exist? So we thought, all right, we'll just start to make it. And that was the kind of the, like many things, it's born out of frustration. But it was like, we should build if the thing that you want doesn't exist in the world, it's time to go out and build it, is what the genesis was. But along the way, we've started to bring in a lot more astronauts, a lot more crews who want to help plan these really ambitious missions. So if any of you want to get involved in the mission, or you have a twin, or you have a spare $1 billion, just come.
Yeah, please.
Yeah, I just wanted to piggyback on that. As one of the summer fellows for BioAstra Crew 2, Chris likes to say he has a very big tent, and anybody who wants to come in and study space flight and space health is welcome, which is I think a little unusual in academia, but very welcome. And so one of the things, I think these Starship missions are great and excellent and very, very interesting aspirational ideas. But one thing that we were able to do, this past summer that is very concrete is take a look at the inspiration for astronaut genomes and take a look and see what's going on in these 4 people who are not among the healthiest humans that have ever lived or flown, as is the case with most of these astronauts. And what is going to be the impact of some of these pharmacological, some of these drugs, things that astronauts take in space regularly. And I think there's a big push to democratize space, to try to sort of, I guess, also increase the tent of those who are doing space flight. And so that needs to necessitates that there will be more health profiles that exist as well that are less than, let's say, ideal or perfect or whatever. And so one of the challenges of either a biobank or trying to look at all of these crews is generating this massive amount of data and trying to understand back on Earth, how does this relate? How are drugs going to work? How does your DNA or your epigenome or your proteome, how are all these things changed for normal people like me and you, and not just the healthiest humans that have ever lived, as we continue to expand and fly more and more people?
Yeah, I'll also add, just in terms of is now the time, I think there's plenty of opportunity to miniaturize and increase our capacity to do a lot of this biomedical research. I think now is the time, particularly because of the interest in new space stations and going on longer duration missions. But if we take the most advanced platform that we've had to study people in space is the International Space Station. That's 1990s technology. And at the time, the Human Genome Project was happening, and that was costing billions of dollars, and you needed machines the size of small rooms to sequence DNA. And now Dr. Kate Rubins, she sequenced DNA on the International Space Station on a device that could fit in your pocket for probably about $1,000. And so if we recognize, how far that type of medical technology has come already, really the opportunity is that we just need to build a facility that has those capabilities in mind from the onset. And the increased interest in having space stations right now means that that's the time. And so if we go from refrigerator-sized machines to something that can fit in your pocket for DNA sequencing, there's plenty of other areas where we've seen that type of advancement. And as long as we can get it in one of these new stations, have the station be planned with these kind of capabilities in mind, I think that makes this the time, regardless of how much more progress can happen.
Amazing. I know we're coming towards the the last half hour of our chat, so I wanted to steer our conversation to why New York. Typically we think of Houston or the West Coast as, or Florida as, being sort of like the leading players in space or space medicine. But what do you see as, the role New York plays in biology and space? Why don't we start with you, Chris?
Sure. And one of them is we can unequivocally say we have the largest biobank that's in actually the first real commercial biobank that's ever existed. NASA, of course, has been banking astronaut samples for decades, but they are not, there's no easy way to get access to those samples. You can, it's often arduous and slow, but not impossible. But it is not something, I mean, we've built the system so you can have a 2-week turnaround time for a decision on access to data, and usually 2-3 weeks for access to samples. Even if the answer is no, you want to know, oh, the answer is no, we don't have enough left. But just want to make something that's, very New York. It's fast, it's ambitious, it gets things done, as it should be, though. Because I think, there was, there used to be part of the data repository that was on NASA's website as early as January this year. They just updated it, but it used to say, allow no less than 26 months for access to identifiable data, and allow no less than 13 months for access to de-identified data, which if any of you have done work in data science and someone says, wait, I have an idea, I want to look at some data, and you go to a website and it says you have to wait at least 13 months before you can access that data, it's depressing, number one, because you have an idea like, oh, okay, I can't use that data anytime soon. And I think it takes away from, I think, the speed with which we need to address these challenges. You can't wait months or years to have access to data, especially if it's been consented, especially if it's de-identified. So I've been pushing a bit to make all of our infrastructure as responsive and as fast as possible, but still be ethical. So all the crew members sign consent forms. They're fully briefed on who's accessing the data, when they're accessing it. But actually, one of the things I've loved about the biobank and the infrastructure, again, it's in New York City. Of course, the most expensive place to put a biobank, but at least it's not New Jersey, I guess. But it is, it's here. But actually, every year we do like a little Christmas card, or like a state of the what happened the past year with their samples and their data. And last year we published a whole compendium of papers in Nature and had this large release of data, and got framed, pictures of the journal to send to the crew members. So I actually view it as we're shepherds of their data and their samples, but I want to show crew members that they're using it similar to clinical trials at the hospital. When any of you, if any of you have signed up for clinical trials, thank you. But when you do, you often would like to get a little email like, oh, did anyone use my blood sample? Is my urine sample just in the freezer? Did someone drink it because they thought it was lemonade? Like, what happened to my samples? You want to know, of course, right? So even any of the All of Us program, or any of you part of the All of Us program, it's the United States. You can get your genome sequence for free if you sign up. Cornell is one of the recruitment sites, actually right by our lab. And there's you get your genome for free, and you can log in and see information about your risk for disease and traits. And so it's we've tried to build it in as the most responsive and useful biobank for astronauts possible. And it's in New York City, but New York State also, up in Ithaca, I think there's the sign for the New York State Consortium for Space Technology. There's a lot of manufacturing here in New York State. It goes without saying, I don't know, it's New York City, so it's got to be exciting by itself, I'd say. And so, well, why New York?
We're big supporters of that. I mean, we build satellites in New York City.
Which is also the most expensive place to build a satellite, I think, probably, right? Yeah.
But it makes sense for us because most of our customers are life science companies and researchers such as yourselves, and they're primarily based in this, Boston, New York, Philly corridor. So it makes sense if, they want to look at our hardware, or if we want to grab a sample, it's a subway ride away. What happens on that subway ride? Anyone's guess, but it's a subway ride away. Devon and Jeremy, anything to add?
Yeah, no, I mean, I was kind of thinking just like, why not? It's kind of how I've always felt about it, that New York is such a hub for so many different facets, so many sectors that are required for space, that I was always surprised that there was such a little space presence here. Frankly, I didn't know, like I came to New York. I'm from New York originally, was living away for a number of years, and ultimately just wanted to come back to New York. And I came to New York and tried to break into the biotech scene until I was connected to Chris, because I had told someone I'm coming from biotech in diagnostics. I just want to work in biotech to like continue this ball rolling. But I'd love to do space research ultimately. And they're like, oh, you got to talk to Chris. And lo and behold, it's Chris is leading, you know, this space medicine wave that's moving forward, and it's happening right here in Manhattan. Yeah, I just feel like the city of New York creates so much connectivity across the world that this sector requires that type of collaboration and just being connected to one another in order to facilitate things. That New York jumps out to me as a place that it makes sense to do this work.
Devon?
Yeah, I think I echo everything you guys have to say, as well as, you know, the broader state of New York as well. So not just the city. The city is great as well, but, you know, New York State has a rich history of being involved in space flight, like Long Island building the lunar lander. And, you know, for our work in space medicine at Memorial Sloan Kettering, you know, as I mentioned earlier, there's 3 particle accelerators within basically striking distance of New York City. Which is hard to say of really anywhere, maybe in the world. I'm not sure. I'm not sure how many particle accelerators per capita are in other places, but it's pretty high here. So for a radiation researcher, it's fantastic. And I think, as Jeremy's alluding to, the talent that I think you can pull, being in New York State, New York City. And I think Empire Space just published a list of essentially space researchers as well, besides us, that are spread out all over the state in Rochester, Buffalo, Ithaca, New York City, Manhattan. So I think New York has a thriving space sector, both in terms of research and kind of technology development. And it's great to see kind of the legacy of the space program being alive and well in New York State. And hopefully we can continue that.
And even one thing I just thought of is that there's we have a project called the Microbe Mineral Atlas we just started, which is trying to find microbes that can absorb and enrich rare earth metals. And one of the places we're looking at are in the old abandoned mines in upstate New York, and looking to see what organisms have adapted there. And so those could be extremophiles that could help us potentially on, you know, on Mars someday. So it's interesting that things that you wouldn't even think would necessarily be spaceflight related, but are part of it because there's just so much of an industrial history in the state that we can use those as lessons for microbes that could help us on Mars.
Yeah, and Jeremy, you mentioned Haven One, Vast. You know, they acquired Launcher, and they were in Brooklyn, and we started in the same room as, you know, when they left, we took over. So there's always, and we were surprised as well. You know, the depth of you know, space companies and space research in New York, you know, at the time. So I guess, you know, before we take some questions, I think what I'd love to, you know, just get a quick, and I know there's a lot of nuance in science, and it's not something that, you know, you throw away as a you know, offhand statement. But I guess, according to you, in the next, say, you know, 3 to 5 years, what would, according to you, be one of the most impactful outcomes from you know, space medicine or biology in space? Devon? You could talk about, you know, the missions that we're working on together, or anything.
Yeah, there's so many things I think in space medicine that can, you know, that can spin out of our ideas today. Everything from, I think, leveraging mRNA technology, like Chris was talking about, not just for the COVID vaccine, but for DSUP and for introducing mRNA to kind of have these interesting new proteins, or you know, like we're trying to do at Sloan Kettering, revolutionize healthcare for people on Earth who are undergoing particle radiation therapy. I think there's a lot of different ways it could go, and I think the law panel talked about some of the kind of technology development that's come out of spaceflight. So yeah, I mean, I'd like to see in the next 3 to 5 years more of an emphasis on, maybe I said earlier, trying to expand the health profiles of those who are going to space, and some of that requires much more genomic data, genomic processing, and things of that nature that are now possible, that were not possible, you know, 15 years ago. And yeah, I think Chris is going to be a better answer to this, but maybe in the next 500 years, we'll start to see maybe some more gene editing and more long-term changes to potentially the human genome to have a more sustainable spaceflight presence in kind of adapting some of our DNA, which is hard to do because it's based on, hundreds of thousands, millions of years of kind of adaptation. But, the next 3 to 5 years, let's just say we could expand the health profile of those who go and continue to improve the health of those on Earth from the technology we develop.
Jeremy?
Yeah, I think something that I'd like to see is maybe more of a perspective shift, hopefully within the next 5 years. Maybe it'll take a little longer, but I feel like the space sector as a whole historically has, certainly has an origin of being coming from a military background. And I think that there was always a sentiment of like accomplish the mission. That's the thing that needs to get done. And I think that attitude has certainly shifted significantly when it comes to the like classical national space programs. But with this new emergence of commercial space, I always say that, companies like SpaceX, they're an engineering company, and their goal is to solve an engineering problem. And that involves keeping someone alive, but it doesn't necessarily involve understanding what's happening to their body when they go into this harsh environment. And so I would love to see more of an emphasis shift towards trying to characterize what's happening to these people who, at the end of the day, are putting their bodies on the line. And I think that because it's such a novel, exciting experience, there are plenty of people who are signing up to go get the mission done. And so that has de-incentivized the necessity for understanding what's happening to these people's bodies. And so I'd love to see that perspective shift. And that means not just, how people talk about it or approach the design of missions, but actually building out infrastructure so that, I don't have to travel across the country in order to make sure that these samples are stabilized. These launchers are building the facilities so that they can study the people that they're sending to space. That's something that I would love to see. I don't know if maybe 5 years might be a little soon, but we'll see.
Chris?
Yeah, I guess I have lots of ideas. I think over 500 years, but I'll stick to just the first few. But, because in hundreds of years, we could have people, sustainably on Mars, maybe at the outer planets, looking at nearby star systems. But in the near term, a similar thing, a perspective shift would be to go to look at life that's in these spacecraft, but also what's going to, because within 3 to 5 years, we should be back on the moon and hopefully staying a bit longer. 30-day missions are what's planned for Artemis. And we should have multiple space stations in orbit by then, actually, even 3 or 4 years, maybe sooner. I've yet talked to VAST. That should be, Haven 1 should be next spring, which is extraordinary. People don't know they're going to put it up there. It's going to actually try some artificial gravity, make sure it works and doesn't explode, and then send crews up there. I mean, multiple sets of crews that go up. And that's next year. And then if that goes well, they'll do more of these Haven launches, which is just really extraordinary. So I'd like to see more of the perspective shift, though, not just the humans, which is a key part that's missing, but have humans then also, in addition, look at the microbes. So we have a project looking at microbial engineering for ecosystems so that plants could grow in lunar regolith, which is currently not that hospitable, or even projects that pull out perchlorate from the Martian crust. And so I'd like to see that the microbial engineering get really good at that point, that we could plant crops on the moon. There's already been some cotton that's been grown on the moon in Arabidopsis, but actually maybe even eat a salad on the moon would be a goal for the next 3 to 5 years. And also just those ecosystems can also help for nutrient recapture, water reclamation, all those systems that are microbial, engineered microbial systems that we take for granted for like fermentation here on Earth, but you could use some of the same tools for survival on other places.
Oh, that's amazing. From a microbial engineering standpoint, that's an outcome that I want to see personally. My 3 to 5 years is use lower level organisms to manufacture materials that just cannot be done here on Earth. So one of our upcoming missions is to produce a novel strain of bacterial nanocellulose, where we think we can expand the existing market here on Earth. Try and use it for novel biofilters or wound care, or maybe in vivo. It is a biocompatible material, and we're working with a partner from New Zealand to try and engineer those things. So our goal, what my preferred outcome is in the next 3 to 5 years is not only do we have these unique materials, but we build out sort of like an engineering and manufacturing, sort of like economy that would directly benefit, Earth and take out some of the, polluting, processes from Earth to space. With that, we have time for some questions. So yeah, would love one. Yeah. Oh, sorry.
Thanks. You mentioned commercial space stations for Starlab or Orbital Reef or Axiom or VAST across three things: regulatory, physical environment, and ConOps. What can they do now to make it easier to do the type of research you want to do? And that's for all four of you.
I mean, one thing they could do that I think VAST has started to do is they put an announcement with SpaceX, for example, with an open solicitation for research ideas. And we've been partnering with SpaceX for the commercial crew since 2021, but also put a bunch of other ideas into VAST. So I think having announcements and say we want ideas of what people have. They can fly for hardware, for algorithms, for mission tools. So that's been good to see. And the regulatory, they're already a bit on the bleeding edge of regulatory because they don't have to follow a lot of the NASA guidelines because they're their own entity. If you dock to the space station, you immediately have to follow all of NASA protocols and guidelines. But if you're a free flyer, which all these stations will be, it's kind of like its own country. So it's really, so regulatory, I'd say be careful. I don't kill anybody, but it's a pretty dynamic space. And then for comms, it's getting better, but it's still like some of the laser-based communications getting ready, so that is early. But you haven't yet had I think, YouTuber in space or a podcast from space, but I think we will. Okay.
One of the things that, I think some of these private space stations should be doing is not fall into the NASA ISS implementation trap, where, if a researcher, is proposing a cool idea, typically what you're doing at the ISS National Lab is you're pairing them up with a implementation partner, which again is a big incumbent, be it Redwire or Space Tango or whoever, and typically what ends up happening is a series of compromises where you don't the outcome of your experiment or your study is not the one you designed. So, I mean, I'm sure, you guys have seen this where, I need a 50 milliliter flask, for example, or a container. The implementation partner comes in and says, oh, actually, I've qualified a 25 one. Can you use that? And then, I need this sensor, but I've qualified that one. So what ends up happening is your end result looks so different to what you actually wanted to study, and you get some results, your experiment is successful, but what does it mean? You don't really know. And so, moving away from that sort of, protocol of, you're a researcher, we don't ask the chef to make their own knife, so here's, Space Tango, or here's, Redwire or whoever, just work with them and they'll figure it out. Instead, it should, I think given that there's so much flexibility with these private space stations, you should be flexible with the kinds of hardware that you can qualify and carry on there as well.
Just to, it might be a bit redundant, but I was just going to say the nature of them being a commercial entity will make the research we're doing easier. And the facility like the biobank that we have now is only possible because we are in this commercial space age where we're working with these commercial crews.
Yeah, I think two kind of well-known stories that I have thought about just over the course of you guys answering. The biology physical sciences section of the NASA Science Mission Directorate is sort of the redheaded stepchild right now, and that's okay. But I think what happens, you hear these stories about biology missions, and they're lauded as successes, and kind of what Shishir is saying is true, where when you start to dig into some of the details, I've heard a story of some Drosophila fruit fly researchers, and I think the biologists take a backseat to the engineering a lot of times because we're often on rideshare. There's no dedicated biology missions, really. You're always kind of lumped in with someone else. And so it's fine that the engineering has to come first, but the engineer approached these Drosophila researchers and said, hey, listen, there's all these air holes for the fruit flies, but they're going to escape, so we have to tape them up before you fly. That doesn't really, they're going to die, right? That doesn't work. And so that's, yeah. So it's an awful example of kind of the engineering taking precedence over the biology, and you don't get a result, however many millions of dollars are spent between the payload development and the launch. And I think likewise, one of the most famous or exciting missions was BioSentinel. And BioSentinel was on Artemis 1 and sat there famously through a hurricane, and they lost power. And eventually, essentially, the payload was dead on the platform before Artemis 1 launched. And there's no way to access it. There's no way to change anything out. There's no way to support the biology. So I think as we develop these kind of private and commercial ventures, I would love to see a full biology mission, or at least more biology voices that have a little bit more of a somewhat equal say at the table compared to the engineering. And of course, that's important. But if you're talking about a biology payload, you need to be speaking with biologists. In my opinion, hot take maybe, but.
Cheers. Hi, everybody. Steve Wood, electrical engineer, patent attorney, and really interested in the intersection of AI, genetics, space, et cetera. So you all are probably more familiar with the fact that once we take the human out of the Earth environment, it will continue to evolve into potentially a different species even. So when you say, Chris, that there may be people in the distant planets, will they be human? Or human derived, or exactly how? And so and I don't want to come across as too much of a eugenicist here, folks, but when we look at some of the experiments that have happened, for instance, the Chinese twins who were genetically engineered to be impervious to AIDS, HIV, and I believe it was some follow-up research at Duke that showed that gene actually imbues them now with more resilient neural tissue, brain tissue, makes denser connections, more resilient to stroke and things like this. And so if we do get to the point where AI is able to design new CRISPR, Cas9, 10, 12, whatever platforms, and it can create new gene sequences in a more perfect human genome, one that would allow us to actually sustain in space long term, because this one won't. Not really. And as you mentioned, the longer you're up there, the more severe your response. And so theoretically, there's a threshold. You won't be able to come back. If children are born on Mars or the Moon, Mars 0.38 G on Mars, 0.16 G on the Moon, and then they spend their formative years there, they'll never be able to come visit us here on Earth. And so with those kind of dynamics in mind, where do you all come down with respect to potentially perfecting human genome for purposes like that, but also understanding that if we then procreate together, those traits may just, permeate our genome? Cheers.
That's a great pitch to read Chris's book, 500 Years, where he talks about all of this. But go ahead, Chris.
Yeah, I wrote a book about kind of along those lines. If you start to go down the rabbit hole of that question, it leads you to a lot of places where you think, well, will people be marooned on a planet if they've lived there long enough? Or even if you just go there for 2-3 years, you can't ever come back to Earth, which I describe as a failure in planetary liberty, is that if you truly have a liberty, you should be able to go back and forth to any planet you want, including either for protection mechanisms or even gene modifications to enable you to come back. So you could ramp up your density, say LRP5, for example, for bone density, ramp up your muscle production, and then come back to Earth, which is something that, again, 5 years ago was complete science fiction. But now we know the targets, we know it's possible, we know some of the spaceflight real likely targets that we could even go after. So I think you could, much like if you're going to go hiking in Mount Everest for 2 weeks, and you could change some of your hemoglobin levels or change some of your response to radiation if you want. Like, what if you could have genes on for the weekend? You're like, oh, it's going to be a crazy weekend. I'm going to be drinking a lot this weekend, so aldehyde dehydrogenase, I'm going to ramp up so I can, drink a lot but not feel like I'm going to die the next day. Which there's actually a company called Z-Biotics that makes a probiotic that does this, which we just ran a clinical trial with, and it was doing okay. And we started looking at the data, but
whole genome editing, you're talking about methylating or unmethylating, phosphorylating.
both, yeah, both, yeah. But you can do both. I think they're both. Epigenetic modification is something that's actually much more plastic by definition and is probably safer, but also is something that might not have as big an effect. So I think you'd have to see what the phenotype you want or where you need to survive, and then match it to the genetic therapy. But yeah, I wrote a whole book about this. Like, why do we have to make all of our? We can't make all of our own amino acids, which makes me kind of sad. Like, why? What happened to our human cells? Why do we have to go get amino acids? We know the pathways to make them all, or even like vitamin C. It has been gained and lost through primate evolution. And like your cats and dogs, they don't get scurvy because they can still make their own vitamin C, but they're mammals. And we have the gene inside of us. It's just been degraded too much, but you could tweak it and turn it back on. So I propose in the book for long-term missions, we should systematically go through every gene that's either still in us and just been mutated, or add a few in so that you could, basically, it'd be easier for resource requirements because you could make most of your own biomolecules that you would need. So yeah, it's a whole, and the ethics behind it, why we do it, how you make sure you're not ableist, and make sure that you have a real context and intergenerational view of the effects of what you're doing. Recommend my own book, I guess, too. Yes. Yeah.
This was quite wonderful. Thank you all. Although I did mention there's not a single female presenting. So hopefully that's an avenue for progress in New York, where we may have female scientists or entrepreneurs speaking. But my question, this is very fascinating and really very inspiring. So Devon, Lundine, Memorial Sloan Kettering. So your research on DNA replication and triple negative breast cancer is fascinating for many reasons. That's quite a difficult area to treat because of the receptors lacking estrogen, progesterone, or HER receptors. But in terms of I'm unclear, are these experiments that you are running that you're planning on running in space, where you're looking at radiation therapy enhancement for cancer patients? So are you actually, since you are based at MSK, are you utilizing cancer cells from cancer patients that you are then transporting to space, where you're looking at gravity? And are these humans, or are these murine models, where a murine model, you still have, I mean, regardless of mentioning that since we're outside of the Earth, we do not have regulatory. FDA, I think, would take a very dim view of that. And Memorial Sloan Kettering's funding is hopefully, we don't know what Robert Kennedy's been up to today, but I hope that gets edited out. But in any case,
I think it's ketamine is what he's been doing today. We think.
But there's, I mean, hundreds of millions of dollars, if not billions, over the years of federal funding. So I think that anything involving experimentation on animals, human cells, you don't have pain issues if they're cells, but the ethical requirements, FDA would take a very dim view if you bypass them, and you do want to enhance radiation for humans. So I believe, but back to the question, are you taking cells from humans at MSK and using them in space for experiments? And have you specifically obtained permission from these humans?
Well, the short answer is no, we are not doing that. No, no, we're not doing that. As much as we would love to, as you said, we do have regulatory bodies here on Earth that kind of dictate what we can and can't do. So, what we are able to do is use model systems. And so our work on the ground and our kind of proposed planned work in orbit is using TK6 cells, which are human-derived, but have been a model system for a long time and have been space-flown to Mir, and I think on the shuttle program as well. So we're using a well-characterized cell system to look at DNA repair and try to understand DNA repair both in orbit and on Earth. And so, at present, we have no plans to send any sort of patient-derived cells or animals or things of that nature. We'd love to, but one step at a time, I guess. One small step for man, one giant leap for mouse kind, maybe. So someday maybe we'll get there, but yeah, for now we work in really well-established systems, and the breast cancer work was all through my PhD, unrelated to the space stuff. But I'm happy to talk to you about that, separately. But they are human cells, human-derived. They're a model system, so they're no. I mean, I guess the permission is inherent in the fact that they're a model system, rather. So we sort of have, the ability to navigate that.
One more question. Yeah.
Microgravity to draw in big pharma to see space as a strategic domain, much in the same way they see AI today.
I mean, we just did one experiment. We flew a plate of lyophilized about 3,000 FDA-approved drugs to see how they respond in spaceflight. We just got that 2 weeks ago, so I think it's recent and it's in my head. It's something that I'd want to see something like that, or more things like that, where it's a systematic analysis of every possible drug, and also slight variations of drugs, and also maybe peptide screens done also in plates in space. So things like that would be useful, because then you can start to see, as more and more people are going into space, or are they, osteoporosis drugs, hypertension drugs, you could start to screen for them in space.
One of the other applications that we're now seeing is what we call high throughput screening, which is to try and take like a library of molecules and expose them to a target tissue or target organoid. One of the big benefits that we're seeing, and that has also been hypothesized about in space, I mean, it's already been done, so it's not really hypothesized, is we're able to bring forward some of the targeting studies. So UCF and UCSD were able to do some of the screening work on organoids, and bring some of the drug candidates to the pipeline faster, to phase 2 clinical studies faster as well. And so that's an area that is seeing a lot more uptake with pharma companies, because they have an incentive to get drugs to market faster. So that's right now, that's a big that's a big draw for them.
Yeah, I'll just add on super quick. I think the sample size problem comes in where we stand right now, in that, you take our biobank, sample size is pretty low, and that's just observational at this point. If we're really talking about pharma, we need to have treatment group and placebo group and blinds, and there just aren't enough people going into space yet that's happening in an effective, significant way. But I think that it just comes to say that the amount of launches, the amount of people going to space in the last 5 years has skyrocketed. If we have more stations and people are going there longer in the future, it's on the horizon for sure. And there are groups that are working to pioneer clinical trials of different drugs in space. Ursa Bio being one of them. So it's certainly a direction things are moving, and there will be opportunity to do it. It's just we're a bit early right now to say, oh, look at this one particular intervention, or look at this one particular pathway that you might be able to. Yeah, or organoid models certainly are more near term. That's going to be, these like the cell experiments that Devon's talking about. We've had the opportunity of flying some human cell lines on space missions, and organoid models are going to be the next stage of understanding what's going on in that regard. And so, yeah, you might start to see those trials coming about sooner in those model systems. But again, we're still a little early on really organoid models as well. It's much closer, but yeah.
I'll just say quickly too, I don't know a ton about this world, but in space manufacturing, I think as another side of it, where companies like Varda Pharmaceuticals are, I don't get paid by them or anything, but I think they're trying to find, the energetics of having microgravity is such that you can produce pharmaceutical products that are unique and otherwise have trouble on Earth. So they wrote a paper about ritonavir having essentially a bunch of side effects when produced on Earth. There's a form of it that gravity, essentially convection, makes it convert to, where in microgravity that particular form is not there, and so the product is much more pure and readily available. So there's this whole pharmaceutical manufacturing side of it too.
But yeah, there should be a big space station full of organoids, like imagine like a bathtub full of jellyfish. It should look like that, I think. Yes.